and know data science. And obviously, I'm not a Pythonista like you guys. Some years ago, I wrote books together with some alumni colleagues in my lab about Python visualization. Then that's myself to share about in the PyCon events. Where I got to know these brilliant people working on promoting open source in Hong Kong and joined them. Though I also have a bit of challenge keeping up with volunteer work during this final year. Anyway, without further ado, let me take you through a brief journey on unraveling mysteries of life by informatics, which I'm passionate about, and share some bits and pieces about interactive Python visualization on web applications that I ran into during my research. Here's a refresher for Biology 101. You and I and other life forms on Earth are built up of cells which contain long chains of polymers called the DNA, built up from paired nucleotide bases as the basic units, the ATCGs, as you probably heard. And for more complex organisms, right from the baking yeast, there are plants and different kinds of animals. DNA is packed into chromosomes in the cell nucleus. If we consider it in the informatics term, we can see chromosomes are fragments of DNA as strings, with characters written in a chemical format. These strings are called the DNA sequences. The DNA sequence encodes the logic of when, what, and of different materials to produce, modify, interact, and eliminate throughout the lifespan of an organism, which in turn instructs on how to build structures, how various cellular machineries to higher biological functions are working. The sequence virtually defines the being of the organism. Therefore, the DNA sequence can be viewed as the blueprint, or indeed, the source code of the life program. You've heard me using the word genome in the phrase whole genome mapping, and that I'm working on genomics, which is the study of genomes. And you probably heard the word from COVID or other news. So what is it? It is the whole set of genetic materials in an organism and being inherited along generations, giving all the instructions for the life program. And by the informatic definition from the previous slide, it is the sequence strings we find in the cell as a full data set. In human, just a non-redundant set we receive from one of our parents contain 3 gigabase pairs, which is 3 times 10 to the power 9 characters. There's a massive combinations for substrings for different functional elements, with the most well-known of all as what we know as genes. Since the early days, when people try to study these genetic materials, there is this molecular technique called karyotyping, which is to align, order, and expand chromosomes under the microscope. Many of the anomalies in the chromosome lead to genetic disorder, such as the extra chromosome 21 that caused the Down syndrome, and the Peter-Shaw syndrome where a large piece of chromosome 5 is missing. Like these two, many of these diseases affect facial features and the progress of development. Many others even have significant impact on the life expectancy of the patients. Although karyotyping has made a still contribution to biological research, karyotypes can be difficult to read even for the much trained eyes, and the resolution is quite low. Changes of at least 5 times 10 to the power 6 unit size can be detected, and often only the much larger ones have a better recall rate. There's no sequence context. We hardly know what inside about the records. So, other techniques trying to improve the resolution, but can still only zoom into like 100 kilobase pair. And they are good as well, meaning a lot of time and resources are needed even just to get some information from a small region in the genome. Foreign sequence knowledge may be acquired for some of the techniques, and that also hinders novel discoveries. That's massively differently in the next generation sequence periods, we can get sequence information from the whole genome down to per base unit level resolution in a single experiment. Since then, the cost of sequencing has been dropping quicker than even the computation cost of the famous Moore's law. And this method is basically about randomly breaking down the genome and DNA molecules into millions of small fragments sized around 150 base pair units, and obtain the sequence information of all of these fragments in high parallelism. The sequence of these small fragments, known as sequence reads, are then overlapped and assembled into longer sequences. You can then orthogonal data in the sequence of the process, like here, the orange lines here, as threads to update of the fragments. Greatly improve the resolution and throughput. It has its caveats, mainly come from the short sequence read length. Let me draw an analogy with playing puzzles. We try to assemble small puzzle pieces back to complete patterns. Spreading rounds of as well as other localized duplications. Our genomes are indeed predominated by many repeating and similar sequences. Another problem is that when generating in the consecutive data tandem repeats, when there's an array of multiple copies of elements, it determines the copy numbers, like on the bottom right corner, if we have a lot of redundant elements, like how many we should put to get the original picture. And these issues result in missing sequence and order information when short fragments of sequences fail to be connected. In science, when looking for answers, we often use it to compare and contrast. We compare samples and see what are the differences and investigate the effects of the differences and how they arise. That's sort of like debugging, isn't it? Just like when you use GitHub to compare two script versions. And variations in genetic materials have various biological implications. They can directly cause diseases or affect the susceptibility and treatment outcome. They can alter the yield and properties of crops and livestock. The understanding in genetic variations can also provide insight into the origin and evolution of various populations. Therefore, proper research will benefit society in multiple aspects. And variations come in different sizes. You can imagine small differences like the single nucleotide variations as changing a single variable name or function in the code, and larger ones called the structural variations, SVs, like removing, inserting, or modifying a large chunk of the script. Of course, depending on what and where it's changing, small differences can still have large impact, as in what differentiates between all these Greek alphabet strings of the COVID virus. Yet, it is pretty obvious how the large changes must not be overlooked. And echoing the title of my presentation, there are two levels how this massive important information not being well studied enough. But despite the importance of the larger space, which are studied to give some properties important to like agriculture, medicine, but they were studied like cut off one by one or a few or a few. So the first one is on the size like larger, but still submicroscopic variations have been hard to study. And before technological developments further advances, scientists could only focus on smaller changes by making the corresponding definitions. They limit themselves to around like over 50 to maybe a few hundred base pairs, and comparing to the whole chromosome only, that is very small. And there are modern definitions like with long read sequencing, and people try to look at like 1 kilobase pair or more, but so far the sizes from 10 kilobase pair up to maybe 10 base pairs, those are still challenging for sequencing technologies. And the second thing that's being neglected is the repeat that I was talking about. There's repeating sequences are what predominates the genome space. Although the protein-coding genes are the most relevant. And much of these repetitive sequences come from the transposons, like the jumping genes. That dominates most of the eukaryotic genomes, including plants and animals. These jumping genes, as they jump around, they copy themselves, and they disrupt the genome structure as already moved. And besides the transposon activities, there are also other errors in DNA replications and repair mechanisms that also involve highly similar sequences. And if we ignore the repeating elements, we cannot really study genome structures well. But repeats have been hard to study because they disrupt the genome graph when we assemble things. And because of the similarity, when comparing, we need high computational power. So they're often masked in routine genome assemblies. So when the sizes matter, another dilemma of losing resolution or losing the big picture, where's the sweet spot? There is this advanced genomic technology called the optical mapping, which generates ultra-long maps up to chromosome length, making it a very long thread sufficient to stitch on more smaller pieces of the sequence fragments. It is called mapping, but not sequencing, because it does not provide a base-by-base information. But why is it still useful? It's like when you look at a map and you just want to find where the one stop. Don't really want the list of what that is at every street number. And this method fills up the continuous gap of linkage information as sizes from kilo to megabase pairs. And with non-math links, the complex and repetitive genome regions, then challenging to other technologies can be standard for us. So here I would like to show you the machine in qualitative generation and a snapshot of the first instantly labeled single DNA molecules and arrays of size channels. And this is a figure I drew for review of optical mapping that summarizes these working principles. I will leave the details for further reading, but the basic idea is that the assembly of genome maps is quite similar to assembling sequences, as we discussed earlier, except that the maps are much longer and they have different data nature. Instead of strings, containing characters, they are numerical values that show the distances between the fluorescence labels. And these longer maps are useful in both train up sequences as well as detecting the large SVs. So by directly comparing in a map-to-map manner. So that's it. The basic idea is to chain more fragments, have a longer thread to get longer contiguous linkage information, and this is the optical map that helps. And yes, I did really help piecing this in genome puzzle. And here are two examples of research studies involved, showing how observing the signal patterns of the optical maps can generate biological insight. But I know you're not here today for biology lecture, so sorry for the non-background. And here comes the meat. And echoing the theme of PyCon HK this year, the data science of the world. Of course, you need to think data how we approach them, not just at the analysis level. And data scientists all know the importance of clean data, so we need to do QC, we need to do cleaning, and finally we want to communicate our results with some in a more intuitive ways. And in the optical mapping, I would say it's a more niche field comparing into the fast sequence data research realm. And there are much fewer tools than we can use as sequence data. And firstly, we are mostly relying on the manufacturer's software, which has a quite complex project structure with dependency on some closed source components and scripts in multiple languages. We're just not so trivial to install and use. And but the key components of analysis, which is the alignments of maps to compare. There are a number of open source tools already. And the gaps I see in this cycle so far is firstly is on the data QC. And currently it's mostly relying on the manufacturer software, which only provide limited QC metrics, and they only use some ballpark total error and average, but not the distribution. And there are some metrics unused, but the data quality is very important, not just to the analysts, but also I'm also working on a REST lab. So for biologists who do the REST lab experiments, they need to know what kind of data are good, so they can stop the experiment and concur on to analysis, or to get another to run another experiment, or the more computational people will think, oh, okay, this data set I get from the database cannot be used, I should get another. And also there, as I mentioned, the repeats are important. That also happens on the map scale. But the repeat analysis is not so developed in optical mapping, and the visualizers are also lagging behind. Like in the previous slide, when I show all my propagation figures, I took a whole long time to polish them in Illustrator, because there are lots of like extra masks that messed up with the figures, etc. And so I'm developed too, and I try to give some more a better POC in a dashboard way to help the practical guidance in experiments and getting public data. And I want to find the repeated regions, like content and copy numbers, etc., and generate more user-friendly visualization. And I was going to say that when we design something, we must think of the end users. And for the users of bioinformatic data, there are quite a few cases of them. There's the most hardcore engineer. They are the ones who designed the most elegant methodology for others to use. And maybe I'm somewhat in between. Like I understand the theory of and carry out both the analysis, some developments, and a lot of experiments. There are also some non-technical, like non-IT biologists who mostly work on experiments, but they also want to know better about the data. They want to present their results. So my design principles is to make the software more both friendly and also more friendly to analysts and developer. So it can be used on both command lines for better performance on automation, and there are also point tracking for people who are used to it, and also because we need the data visualizations. And I want to design it in a structure that can facilitate the further development. So one of the examples is that, like in the manufacturer's software, I find an interesting property, like whether history for the single molecules you see the lines, the blue lines, they are maps. And when they are assembled, they are still old maps of similar properties, just without the variable or optical or signal noise ratio. So I don't quite understand why we can't use similar functions on them. So I want to build it with data classes to reflect the intrinsic properties of the data instead of formats of the data. Yeah, I can let you have a look at this format for the shorter maps, like the unsampled one. They have the signal the summon link. Yes, they have the summon link in bundle. So and two modules. And for us, they have no position in that. So for some conversion we also need, and I create some like operation specific to this type of data from handiness and some statistics and metrics. And yeah, to build this software, we have all these choices. Like in Python, I can choose to a Linux application in PyQt. You can enter the web UI like something like Django and Flask framework, and but there are also these already developed like interactive data visualization and dashboard framework, mostly like Flowy, Bokeh, and all of these ecosystems. And I would definitely prefer using the one-stop solution of the dashboard framework than to recreate, reinvent wheels for everything, because I only want to get the final. Yeah. So dashboard thing is a current trend in data science this year. They handle and visualize data in streamlined ways, and clients can to do something and look at the different aspects of this. And also very looking your themes developed by the community when you are in, using the more used. And for like why I actually choose the Plotty and Dash. That's when I compare Plotty and Bokeh. Plotty has a larger community. And that's abstraction because for my very niche data type, I need a lot of customization and yes, and also that's why Plotty comparing into like both the Bokeh and Plotty's ecosystem, the community is larger. And yeah, here's the basic web structure. Also, by some time around, I also think that the flatter structure is easier for terms of and callbacks. And yeah, that's what I did. Yeah, and when I will start writing this software, like I also wonder like these there are like all these Dash components are imported from different packages. But yeah, it's quite easy to use. Like when just as you write an HTML, like just put all those components as you know about the text. And like very recently in this month like Dash 2.0 is here, and there are quite some breaking changes. Like there is no longer like import A from B, import C from D. At least most of them like they only mention a few, but there are actually quite some more of these import or thing from different types. They also have the different syntax. But one thing to note is that for the Bootstrap components, the import Dash Bootstrap components as DBC still not change. You still cannot import DBC from Dash. Yeah, and yeah, they're getting in some
it seems like that the internet connection from Claire Chung is not so good. We might just wait for a moment and hope that
hello, hi.
Oh.
Karyotyping is come back. I just call him. Just call her.
Oh, good. Very good.
Yes, sorry for all the internet connection problem. And yeah, I will quickly wrap up things up.
Yes. Okay.
Yeah.
And Claire, could you go back a bit earlier? I think. Here, yes.
Oh, yes. I think and yes. Sorry for the disruption. And we were talking about the new dash 2.0, where there are some frequent changes from not just the backend, but how we use the package, including in the fact that we no longer need to import different components from a different larger package, as to also provide a lot of lines. Right? And actually, there are like more lines than they show in this example. But we need to take note that Bootstrap components that gives us the built-in menu that is responsive with Bootstrap have already in the DBC. We still need to use the old way, import hot as DBC instead of from dash import DBC. And yeah, they are giving us this syntax deprecation notice already. So we are going to change. And for the Plotly and dash the same. There are another product company. The streamline of having Polyplots into the dash is quite smooth. Also, in Plotly we have all these components like lines and shapes as traces, and the traces are drawn into figures, and the figure can be directly put into the dcc.graph component to be used in that, just like those diff or some like HTML elements, and that's helpful. And there are also some nuances. Like I asked a silly question on Stack also earlier when I first started the project, like what's the difference between like hash trace and pen trace? So sometimes we do need to pay attention of these similar word functions. So one is adding increase by trace, but does not return the figure. So when we want to store and recycle something, and we need to use the one that returns the figure. And actually, not just they updated to the 2.0 in August. I actually started the project August earlier, and then who knows that they actually keep changing things. And they were announced collaboration with Holofields in January this year. And actually, even after that announcement, on time there was quite a period of time that I tried that somehow the Poly Express histogram was slower than having Holofield's histogram plotted to Poly Dash. I don't know why, but now it's not the case. So I'm going to update that. And another breaking change in the Dash 2.0, and you see here like for interactive visualization, we need to know how the app is getting data from our like interaction. And so there's this output and input, and somehow out for like input that is also output, they need to be put into some import state. At least that's true for the Dash 1.0, and they're all in positional arguments. So I was putting comments to count all the arguments so I don't mess things up. But there's a tower here. And also like because like interaction is so much effort, like that's how open source can make it so they easy like for some more infrastructure things. And for there's another thing, they also update a lot about callbacks. So instead of calling him back, declaring the decorator as the app you install or the declared, and they can also directly from Dash unless you have multiple app in your project. And yes, we can now not choose not to use just the positional arguments but keywords, and if you feel that's safer. And that there's also like they also now encourage us to not use the state arguments, but put everything as input. So we future proof we are going to change that as well. And an example of some features I have wanted but have not seen yet is that the toolbar of plots there give us they only have the native download as PNG, but not the effective graphics that will help publication. So I need to write callbacks for the download. But otherwise, like they handled all the zoom in and out nicely, so I like that. And yeah, I guess since I don't have the time to demo unless you guys wanted, or I can ask any questions. And like I'm still doing, I need some performance optimization, benchmarking, and the GUI design is not complete yet, but I'm going to do that. And I'm also for future perspectives, I'm considering GPU consideration because the maps are getting longer, there are more signals, and that is more data intensive. So here's the take-home messages. Well, as data scientists, we always think of the whole data cycle to fill in gaps, and we need to think about our target users and choose the right tools. That may only be true for the current time, like about Python dashboarding and like everything is so fast moving, but at least it's improving in the good way. And yeah, I like genomics. Yeah, thank you. Let's see.
Okay. We are actually a bit late, but we might take one question. Anyone?
Are there any questions?
So.
Hi, hello, Gary.
Yes.
Hi, thank you for the talk. Yeah, hello. I heard you mention that the Docker file was huge. I wonder, what do you know? What was the reason that those libraries has a big Docker image? In one of your slides, you mentioned the Docker is huge.
Oh, okay. Well, I think they mean to make things easier, so you just Docker install now and things are done supposedly. But then for that package, like it's a whole system that's not just for analyzing, it's also for data management and managing different projects. So it's not very end user oriented. It's kind of like the project manager oriented stuff. They provide they have a commercialized server that have this software installed. Of course, we can still install it locally as end users, but sometimes it's a bit overkill, and we only need to use some of the components instead of all the whole size. And why it's so large? Because they also input some large reference files that are actually the data, but not the software. That makes it like 12 GB.
I see. Thank you. Yeah.
Okay, so I think that's about it. Yep.
Yeah,
so if you want to ask any question, I think Claire usually appear in community events, and you can find her to discuss more. All right. So thank you, and we might have a 5-minute break, and then we will have a very last speaker from the UK, and yep. And so I might see you all in 5 minutes.